Drug-resistant tuberculosis, starting with the definitions. So mono-resistant tuberculosis, mono means one. So resistant to any first line anti-TB drug, which is your HRZE. Poly means many. So more than one anti-TB drug, but it should be other than H and R together, like combination of pyrazinamide or rifampicin, pyrazinamide or ethambutol. Isoniazid resistant tuberculosis definition itself says resistant to isoniazid. Rifampicin resistant means resistant to rifampicin either phenotypically or genotypically. Also, they are now classified under an MDR-TB. If it is resistant to isoniazid, that is Z, H, and also resistant to R, it is called as multi-drug resistant tuberculosis. If it is resistant to H plus R plus fluoroquinones, then it is pre-XDR TB. If it is resistant to H plus R plus fluoroquinolone plus your bedaquiline or linezolid or both, then it is termed as XDR-TB or extensively drug-resistant tuberculosis. WHO has grouped medications into A, B, C. If we are not able to use any of the first line anti-TB drug, we have to pick one of the drugs first from the group A, then from group B, and then from group C. So remember, group A has four drugs: levofloxacin, moxifloxacin, bedaquiline, and linezolid. We have to use all of them in cases of MDR-TB. Again, if you're not able to use these medications, we group to group B medication, in which we use both these drugs, which is clofazimine and cycloserine. If not, then we have to use ethambutol, delamanid, pyrazinamide, imipenem and cilastin or meropenem, amikacin or streptomycin, ethinomide or prothionamide. When these drugs are not used, we can use these drugs from this box. Some more definitions. Extensive TB is when we call a lung which has bilateral cavities or consolidation. Severe EPTB is when a patient is suffering from TB meningitis, CNS tuberculoma, spinal or skeletal TB, or disseminated TB. Severe extrapulmonary TB under a 14-year-old child is any other TB other than the tubercular pleural effusion or a TB lymphadenitis. Bacteriologically confirmed TB is when a patient has Zn staining positive, culture positive, which is your BACTEC culture, and your CB-NAT positive. Drug susceptibility testing is done in vitro using phenotypic method, meaning that disc-Kirby method, where they have a zone of inhibition. Then drug resistant testing, which is nothing but your CB-NAT, which detects your genotypic method molecular techniques. It uses the principle of PCR. If you look at the CT scan, this is a coronal section and this one is the axial section, showing multiple nodules, which is millet shaped, randomly placed in the bilateral lungs in all the zones. This is termed as miliary tuberculosis. And interestingly, miliary tuberculosis is coming under the disseminated TB, the definition of which says the presence of TB in two or more non-contiguous sites, meaning it could be the lung and it could be the abdomen, meaning non-continuous sites, resulting from hematogenous or lymphatic dissemination of Mycobacterium tuberculosis, occurring as a progressive primary infection like Gone complex, or reactivation of a latent focus and spread, or from a iatrogenic origin. Miliary TB is a form of a disseminated TB. Finishing this slide, this class with the epidemiology, that is the burden of drug-resistant TB globally and in India. India is accounting for about 27% of world MDR or rifampicin resistant TB. If you look at the global incidence of newly diagnosed TB. The MDR TB is about 3 to 4%, and in that 3 to 4%, 18 and 21% are the ones who are previously treated for TB.